Excellent. All right, and I will invite our next presenter up, which is Jersey Fontz. Excuse me. So Jersey Fontz is a PhD student in the Waterloo Wetland Lab, spearheaded by Dr. Rebecca Rooney at the University of Waterloo. Jersey's project involves using trait-based ecology to promote the active revegetation of long-point wetlands after the herbicide-based suppression of non-native Phragmites australis in these areas. Jersey is hoping to help the native seed bank by creating and adding seeding prescriptions onto them to bolster their natural biotic resistance against Phragmites and other non-native species. So I know Rebecca touched on this a little bit, so really excited to learn more. So I'll let Jersey take it away. Thank you.
Awesome. Just confirming that you guys can see my screen.
Yep, looks great.
Cool. Awesome. Thank you so much for that introduction. So today I'll be talking about leveraging priority effects to resist biological invasion in marsh organs. And as Rebecca commented on a couple of my topics, I just wanted to reiterate a couple of them. So biodiversity in wetlands is threatened due to the spread and invasion of non-native plants such as Phragmites australis, as many of you know. So to suppress Phragmites in highly productive wetlands, such as those found in Long Point and Rondeau provincial parks, the Long Point Emergency Use Registration Pilot Project was implemented in 2016, where a glyphosate-based herbicide was applied to chosen areas where Phragmites was growing, such as those as you see in the picture on the left. Previous monitoring work from students in our lab found that Phragmites was effectively suppressed in treated areas, such as what you see in the picture in the middle. However, an unwanted consequence in the years directly after such successful Phragmites suppression is what you see on the picture on the right. Some treated areas were then dominated by other non-native species, such as European frogbit. Further monitoring has found a resurgence of native plants after these initial conditions. This is a graph of the average water levels of Lake Erie since the beginning of the suppression in 2016. So the monitoring results coincided with a period of relatively high water levels in Lake Erie, which is what you see kind of in the middle, leading some to hypothesize that these conditions were able to limit Phragmites growth because something similar was seen under greenhouse conditions. But as the water levels are now predicted to fall, which is what you can see here from January 2021 to 2023, we don't know for certain how the vegetation will respond. So our work employs what restoration ecology likes to call active restoration of vegetation rather than passive restoration, which is what was previously implemented after the herbicide suppression. One of the reasons that passive restoration might not be adequate is that the influx of native seeds, either from the surrounding areas or the seed bank, is insufficient to revegetate the area, or that the influx of native seeds or their propagule pressure is too high for the native species to overcome. Active restoration then calls for applying methods into restoration, which would help target vegetation, so in my case, a diversity of native vegetation to flourish. And this can be achieved, for example, by seeding with native species. Active restoration can be successful because seeding native plant species before non-native plants have the chance to establish and grow can give the native plants a competitive advantage through priority effects, which is what we can see here, meaning that a benefit is conferred to the species that arrives first. And this can be through many mechanisms. The example here is through niche preemption. So for example, we can see that the gray triangle species has arrived before the blue or yellow triangle species, leading to that triangle species niche being filled, and thus the resulting population only has gray triangle species. So in my case, I guess that would be a diverse array of native plants rather than non-native plants who occupy the similar niches. So our objective was to test the effects of seed mix diversity, seed mix density, and Phragmites propagule pressure on floristic quality, native and non-native relative abundance, and above ground biomass at a reduced water depth using marsh organ experiments. And the results of these objectives are the ones I will be talking about today. We chose to use marsh organ experiments as they provide advantages in terms of realism over more controlled greenhouse experiments, such that they allow for the study of the influence of the reduced water depth, which is what we can see here, since the plants are growing at a higher level than where the water is, without other confounding factors, while still maintaining this in situ experiment such that the plants are growing under variable conditions of temperature and light intensity. And it's called a marsh organ because they are made of pipes that altogether look like a pipe organ in the marsh. And so we hypothesize that a higher seed mix diversity is going to increase floristic quality, as well as native species relative abundance and above ground biomass relative to non-native species within the marsh organ pipes, and that a higher seeding density will also increase floristic quality, as well as the relative abundance and above ground biomass of native plants relative to non-native plants. So in May of this year, we went out to Long Point and constructed these marsh organs. Each organ either contained soil from a treated or a not treated or control area, representing a low versus high source of Phragmites propagule pressure. Each organ either contained also a high diversity of seeds, a low diversity of seeds, or no seed mix at all. And it was either also a high density of seeds, a low density of seeds, or once again, no seed mix at all. As it shows on the table on the left, the high diversity seed mix contained 15 native species, while the low diversity seed mix contained 4. These 4 species were also represented in the high diversity mix. The seeds were locally sourced from Ontario Seed Company based in Kitchener and rapid cold stratified to break their dormancy, as we were planting them in May and hoping to see some growth by the end of August. At the end of August, we harvested the above ground biomass. From these data, we calculated the relative abundance of each species, the standing crop biomass in each pipe of each organ, as well as the floristic quality, species relative abundance, and compared them between the different treatments. There were many, many results, many, many graphs, and I'd like to highlight some of the key ones. So firstly, we took a look at the effect of seed mix diversity, density, and Phragmites propagule pressure on FQI. FQI, or the Floristic Quality Index, is a biological indicator that reflects upon wetland integrity and the outcome of restoration efforts. A higher score usually demonstrates a better outcome. And FQI can be represented in many ways. We just chose to calculate it as the mean coefficients of conservatism. And from our preliminary results, we can see that mean CCs, or mean coefficients of conservatism, are greater than the control in pipes of any of the treatments. And this is represented by the purple and blue bars being higher than the gray bar. This means that there was a better restoration outcome with the addition of any seed mix, whether at a high or low diversity or high or low density, on soil that contained a high Phragmites propagule pressure. Specifically, if we look at the graph on the left, we can see something interesting in that there seems to be more variability in this restoration outcome when either diversity or density is high, but its partnered variable is low. So for example, there's more variability in the outcome in the left graph when the seed mix applied is at a high density, but instead at a low diversity. We then took a look at relative abundance and quantified that by counting individual plant stems. And we found that native species abundance was greater than the control when using a high diversity mix, regardless of its density. And this is represented by the purple bars being greater than the gray bars in both graphs. However, there was only a difference between the low diversity seed mix and the controls when it was applied at a high density. And we can see that in the right graph. If we take a look, we can see that the level of diversity here then plays an important role in native plant relative abundance. And then finally, we took a look at above ground biomass, which we quantified using dry plant weights. And we found that impressively, non-native above ground biomass was suppressed with the addition of any seed mix, regardless, once again, of density or diversity level. So for example, in the graph on the left, we can see that non-native species dry weight was greatest at approximately 14 grams in the control, while both treatments had non-native dry weights greatest less than 5 grams. So in terms of next steps for this work, as Rebecca mentioned, we want to run some analyses to confirm the statistical significance of some of these general trends represented by the graphed results. And then from these analyses, we can then recommend which combination of density, diversity level, which would be most appropriate for active restoration projects based on the feature that they're trying to control. So for example, our preliminary results show that a low diversity, low density seed mix is enough to be able to suppress non-native plant biomass by greater than 50%. But then the trade-off might be that relative abundance of native species might not be as high if you used, say, a high diversity seed mix. These recommendations can then feed into some future work I have coming up, which involves increasing the scale from the small growing area that marsh organs have to 100-meter squared plots of various water depths. And that's everything I have for you today. I want to thank the University of Waterloo, Canadian Wildlife Service, and the Ministry of Natural Resources and Forestry who have made this project a possibility. A special shout out to all the members of the Waterloo Wetland Lab for their amazing support during this project. And finally, I want to thank all of you for listening to my presentation.
Thank you very much, Jersey. That was excellent. Great to hear more details on those marsh organs and how they were working out. I'd ask if anyone has any questions to just submit them in the Q&A, and we can also submit them in the chat later for Jersey to answer. And I just had one quick question for you. Is the marsh organ, is that like a typical instrument used in these types of studies, or was it sort of new to this Phragmites study?
Actually, it's actually pretty new. One of the previous studies in our lab was done by Megan Jordan, who also used marsh organs to do Phragmites work. But it's mostly a coastal research tool that they use to be able to see how growing differs based on different water depths. So for mine, I only did a pipe height of 33 centimeters, so 33 centimeters above the water level. But if you see other examples of marsh organs, we can see them at various levels.
Very cool. No, it was excellent. And there was a bunch of questions that just flooded in, of course. So is there a benefit to sourcing seeds from nearby sites?
That's a really great question. I would say that there's a trade-off there in that you are making sure that you're not introducing anything that while being native isn't really found in that specific area when you're doing all these restoration projects. But then there is the idea of over sampling of specific native plants and then creating communities that are not representative of a greater diversity, but of the seeds that you are able to find.
Gotcha. Excellent. What non-native species were you watching for? This will be the last question.
Specifically, what we found growing was there was Phragmites, there was also purple loosestrife, and I'm pretty sure those were the two most dominating ones.
Excellent. Well, great. Well, there's a lot of questions for you that we'll put in the chat. So thank you for reaching out that way. So thank you, Jersey, for the presentation. And we'll move on to our last component of the science update.